When transcription occurs to produce RNA, it does so in the 5' to 3' direction using the template strand of DNA. What results is an identical copy of the coding strand, except RNA uses U's instead of T's. Then, translation occurs in the ribosome, and each codon, a group of 3 base pairs, codes for an amino acid. We can translate an mRNA strand into an amino acid sequence, which is what forms protein, of which there are millions of types with various functions. For the coding strand above, I transcribed it into an mRNA strand and then translated it into the protein below using the coding dictionary. AUG is methionine, the start codon, and UGA was the stop codon. Sometimes mutations can occur either during DNA replication or during transcription. One common type of mutation is a point mutation, which occurs when one base in the DNA sequence is substituted for another. When this substitution occurs between two purines or two pyrimidines, it is referred to as a transition, whereas a substitution between a purine and a pyrimidine is called a transversion. So, a change between an A and a G would be a transition because they are both purines, but a change from an A to a C or a T would be a transversion because C and T are pyrimidines. There are several different types of point mutations. The first is a missense mutation, which causes one amino acid in the sequence to change due to a change in the codon. This may or may not cause a change in the protein's function, depending on how similar the two amino acids are. In this example, the A is changed into a C, which encodes for threonine instead of asparagine. This is an example of transversion. A nonsense mutation causes a shorter amino acid chain due to a codon being changed to a premature stop codon. Here, this G is changed to an A in a transition. This causes the codon to become a stop codon, which severely affects the protein's function. A silent mutation occurs when the base that is changed in the codon, normally the third base, does not lead to the coding of the amino acid to change. This is shown here where this U is changed to a C in another example of a transition. Because GCU and GCC both code for alanine, there is no change in the protein's function. Finally, a read-through mutation is when a stop codon is changed to an amino acid. This causes translation to keep occurring until it reaches another stop codon, leading to a much longer protein than expected. Here, a transversion occurs from an A to a C, causing the stop codon to become cysteine. Then, translation continues for proline and glycine, and it continues beyond that as well. We do not know how long the protein extends for, but we can assume that there will be a significant change in its function.